Okay. Chapter 13, Molecular Bases of Inheritance. When T.H. Morgan's group showed that genes are located on chromosomes, the two components of chromosomes, DNA and protein, became candidates for the genetic material. The key factor in determining the genetic material was choosing appropriate experimental organisms. The role of DNA in heredity was first discovered by studying bacteria and the viruses that infect them. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. He worked with two strains of a bacterium, one that was pathogenic and one that was harmless. When he mixed heat-killed remains of the pathogenic strain with living cells of the harmless strain, some living cells became pathogenic. He called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation of foreign DNA. So here we have the S cells, which are the pathogenic ones. When they were injected into the mouse died. The R cells were the harmless ones. When they were injected into the mouse was healthy. They heat-killed the S cells, so they killed the pathogenic cells, injected them into the mouse, and the mouse was healthy because the cells were dead. When he mixed these heat-killed S cells with living R cells, those living R cells took up genetic material from the pathogenic dead cells, and as a result, the mouse died. Later work by Oswald Avery and others identified the transforming substance as DNA. Many biologists remained skeptical, mainly because little was known about DNA and they thought proteins were better candidates for the genetic material. More evidence for DNA as the genetic material came from studies of viruses that infect bacteria. Such viruses, which are called bacteriophages or phages, are widely used in molecular genetics research. A virus is DNA or RNA enclosed by a protective protein coat. Viruses must infect cells and take over the cell's metabolic machinery in order to reproduce. So here's a bacteriophage that is infecting a bacterial cell. The phage head houses the DNA or the RNA. The tail sheath contracts and pushes the DNA or the RNA into the cell. The tail fibers allow the virus to dock onto the bacterial cell. In 1952, Alfred Hershey and Martha Chase showed that DNA is the genetic material of a phage known as T2. To determine this, they designated an experiment showing that only the DNA of the T2 phage and not the protein enters an E. coli during infection. They concluded that the injected DNA of the phage provides the genetic information. Okay, so batch 1, they radioactively labeled the sulfur. Sulfur is only found in protein. It is not found in DNA. So they allowed the bacteriophage to infect the bacteria, and then agitation freed the outside phage parts from the cells, and then they ran it through a centrifuge, and the centrifuge, after it spun, it formed a pellet at the bottom. So the pellet at the bottom represented the cellular material, and the liquid that was on top of the pellet was the stuff that remained outside of the cell. So what they saw, what you can see here is the pink, the sulfur remained outside of the cell. So phage protein did not enter the bacterial cell. Now, their second batch, they radioactively labeled phosphorus. Phosphorus, as you know, is found in DNA. It's found in the sugar phosphate backbone of the DNA. So they allowed the phage to infect the bacteria. They ran it through a centrifuge. A little pellet formed at the bottom, and the pellet was radioactively labeled. Okay, so what that told them was that it was the DNA that entered the bacterial cell. So they concluded that it was the DNA that was the transforming agent, the genetic material. Okay. Additional evidence that DNA is the genetic material. It was known that DNA is a polymer of nucleotides, each consisting of a nitrogenous base, a sugar, and a phosphate group. In 1950, Erwin Chargaff reported that DNA composition varies from one species to the next. So here is one strand of the DNA molecule. It's an alternating backbone of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. These are the nitrogenous bases. Notice that thymine and cytosine, they have a single ring. They are your pyrimidines. Adenine and guanine have a double ring, and they are your purines. Notice that DNA has a 5' end and a 3' end. That deals with the numbering of the carbons on the sugar. So you always start to the right of the oxygen and count clockwise. This is carbon number 1, 2, 3, 4. The 5th one is off the top. Now, the end of the molecule is the 3rd carbon with an OH attached to it. So that's the 3' hydroxyl end. Versus up here, the last part of the molecule, the phosphate, comes off of the 5th carbon of the deoxyribose. So this is the 5' phosphate end. Two findings became known as Chargaff's rules. The base composition of DNA varies between species, and in any species, the number of A and T bases is equal, because adenines always bind to thymines, and the number of G's and C's are equal, because guanine always binds to cytosine. James Watson and Francis Crick were first to determine the structure of DNA. Maurice Wilkins and Rosalind Franklin were using a technique called X-ray crystallography to study molecular structure. Franklin produced a picture of the DNA molecule using this technique. Here she is, and here is her photograph of the DNA. Franklin's X-ray crystallography of DNA enabled Watson to deduce that DNA was helical. The pattern in the photo suggests that the molecule was made up of 2 strands forming a double helix, like this one right here. This is a double helix. Notice that the A's bond with the T's through hydrogen bonding and the G's with the C's. Notice also that your pyrimidines always bind with a purine. Notice that the structure is anti-parallel. Here we have a 5' over here, we have a 3' over here. So this strand is upside down compared to this one. It is anti-parallel. Franklin had concluded that there were 2 outer sugar phosphate backbones with the nitrogenous bases paired in the molecule's interior. Watson built a model in which the backbones were anti-parallel. Their subunits run in opposite directions. At first, Watson and Crick thought the bases paired like with like, but such pairings did not result in a uniform width. Instead, pairing a pyrimidine with a purine resulted in a uniform width, which was consistent with the X-ray crystallography photographs. Okay, so 2 purines would make the molecule too wide, 2 pyrimidines would make it too narrow. Purine and a pyrimidine was consistent with the X-ray data. Watson and Crick determined that adenine paired only with thymine and guanine paired only with cytosine. The Watson-Crick model explains Chargaff's rules. In any organism, the amount of A equals T and the amount of G equals C. So here are here's the base pairing in DNA. Adenines with your thymines, forming 2 hydrogen bonds. Guanines with your cytosines, forming 3 hydrogen bonds. Watson and Crick noted that the specific base pairing suggested a possible copying mechanism for genetic material. So here's the parental molecule, separation of the strand, formation of complementary strands. So this is the semi-conservative model. Since the 2 strands of DNA are complementary, each strand acts as a template for building a new strand in replication. In DNA replication, the parent molecule unwinds, and the 2 new daughter strands are built based on base pairing rules. Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have 1 old strand and 1 newly made strand. Competing models were the conservative model, the 2 parent strands rejoined, and the dispersive model, each strand is a mix of old and new. So here with the 3 models, we know that the semi-conservative model is the correct one. Okay, so we have this DNA, it unwinds and unzips. The light blue is the new DNA. And then when these unwind and unzip, this is what we end up with. Experiments by Messelson and Stahl supported the semi-conservative model. So they started with DNA that was cultured in a medium of heavy nitrogen, so just N15. And then they transferred the bacteria to a medium with light nitrogen. So what happened after the first round of cell division, they ended up with this band right here, okay? Because if we look here, this is the semi-conservative model. After one round of cell division, you'll have DNA that is half heavy nitrogen, the parental strand, and half light nitrogen, which was what the bacteria was transferred to. After 2 rounds of cell division, remember these unwind and unzip, and you'll end up with 2 molecules that consist of 1 strand of old DNA and 1 strand of new DNA, but you'll also end up with 2 DNA double helixes, which are all light nitrogen, because one of these light nitrogen strands was the template for the second round of cell division. So that supported the semi-conservative model theory. The copying of DNA is remarkable in its speed and accuracy. More than a dozen enzymes and other proteins participate in DNA replication. Much more is known about how this replication machine works in bacteria than in eukaryotes. Most of the process is similar between prokaryotes and eukaryotes. Replication begins at particular sites called origins of replication, where the DNA strands are separated, opening up a replication bubble. At each end of the bubble is a replication fork, a Y-shaped region where the parental strands of DNA are being unwound. Okay, so here is your replication fork. It looks like a fork. So you have helicase. Helicase is the enzyme that unwinds and unzips the DNA. You have single-strand binding proteins that bind to and stabilize the single-stranded DNA. And then you have topoisomerase, which is the enzyme that relieves the strain caused by the tight twisting ahead of the replication fork by breaking, swiveling, and rejoining DNA strands. So notice here that we are unwinding and unzipping in this direction to the left. Okay. Remember, DNA replication can only occur 5' to 3'. So this strand can be made continuously as we unwind and unzip. So here you can see a replication bubble. This is a prokaryotic cell. Remember, prokaryotic cell, circular chromosome, versus eukaryotic chromosome, which is linear. You can have several replication bubbles along one DNA strand that increases the speed and efficiency of DNA replication. DNA polymerases cannot initiate synthesis of a polynucleotide. They can only add nucleotides to an already existing chain base paired with a template. As a result, what needs to be synthesized first is the RNA primer. The enzyme primase starts an RNA chain from a single RNA nucleotide and adds RNA nucleotides one at a time using the parental DNA as a template. The primer is short, 5 to 10 nucleotides long. The new DNA strand will start to form the 3' end of the RNA primer. Enzymes called DNA polymerases catalyze the elongation of new DNA at a replication fork by adding nucleotides to the 3' end. Each nucleotide that is added to a growing DNA consists of a sugar attached to a base and 3 phosphate groups. The difference is in the sugars. DATP has deoxyribose, while ATP has ribose. The anti-parallel structure of the double helix affects replication. DNA polymerases add nucleotides only to the free 3' end of the growing strand. Therefore, a new DNA strand can elongate only in the 5' to 3' direction. Nothing happens 3' to 5'. It's always 5' to 3'. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. So this is like the original picture. Okay, so we're unzipping and unwinding right to left. So one strand, this is going to be the leading strand because it's being synthesized 5' to 3'. Now remember, this is the RNA primer. This has to be made first in order for DNA nucleotides to be able to be added to the end of it. Okay, because DNA nucleotides can't get added to a template. They can only get added to the end of an already growing strand, which in this case is the RNA polymerase. The RNA primer. So this is the leading strand. It gets made continuously 5' to 3' as the molecule unzips and unwinds and unzips. It is made continuously. The problem is the other side. So the other side, as it unwinds and unzips, we would be making it 3' to 5'. That doesn't occur like that. DNA replication only occurs 5' to 3'. So this other strand over here, this is going to be the lagging strand. Okay, so this is the leading strand, this is the lagging strand. As we unwind and unzip, we make a primer and make a little short segment 5' to 3', unzip a little bit more, another primer, short segment 5' to 3', unzip a little more, primer, little segment 5' to 3'. Those are called Okazaki fragments. Now the other side of the DNA molecule is unzipping in the opposite direction. So the two strands flip. This now becomes the leading strand because it's being made 5' to 3', and this becomes the lagging strand because it's made in the opposite direction, 5' to 3'. It's made opposite of the direction of the unzipping of the DNA molecule. To elongate the other new strand called the lagging strand, DNA polymerase must work in the direction away from the replication fork. The lagging strand is synthesized as a series of segments called Okazaki fragments. After formation of Okazaki fragments, DNA polymerase I removes the RNA primers and replaces the nucleotides with DNA. The remaining gaps are joined together by DNA ligase. Okay, so here this is showing the formation of Okazaki fragments, and then the primers get replaced with DNA, and DNA ligase closes the gaps. So more Okazaki fragments. This is the same diagram. Okay, so here you can see zoomed in is right here. So this is our leading strand, which is made 5' to 3'. These have to get made in the opposite direction in order for the strands to be made 5' to 3'. Leading strand, lagging strand. DNA polymerases proofread newly made DNA, replacing any incorrect nucleotides. In mismatch repair of DNA, other enzymes correct errors in base pairing. DNA can be damaged by exposure to harmful chemical or physical agents, such as cigarette smoke and X-rays. It can also undergo spontaneous changes. In nucleotide excision repair, a nuclease cuts out and replaces damaged stretches of DNA. So here we have a damaged stretch of DNA. Nuclease cuts it out. DNA polymerase replaces it. And DNA ligase fuses the gaps, closes the gaps. Error rate after proofreading repair is low but not zero. Sequence changes may become permanent and can be passed on to the next generation. These changes or mutations are the source of the genetic variation upon which natural selection operates. The usual replication machinery cannot complete the 5' ends of daughter strands. Repeated rounds of replication produce shorter DNA molecules with uneven ends. So here we're looking at telomeres. Eukaryotic chromosomal DNA molecules have specific nucleotide sequences at their ends called telomeres. Telomeres do not prevent the shortening of DNA molecules, but they do postpone it. It has been proposed that the shortening of telomeres is connected to aging. Okay, so that's the part that's sort of glowing here. If chromosomes of germ cells became shorter in every cell cycle, essential genes would eventually be missing from the gametes they produce. An enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. Telomerase is not active in most human somatic cells.